Okay, so 7.3, this is the citric acid cycle, also called the Krebs cycle. So a similar thing's kind of going on here where this slide right here is kind of like a big thing to understand from it. Meaning, like, I'm going to show you like all the details here. Really, this slide, if you could, this is like kind of really the level of detail you were shown in freshman bio. Like, they didn't show you like the actual chemical structure of everything, but this would be where like this is kind of gets into like the inputs, the outputs, and the general understanding of what you're doing with the molecule. So I'll go back to that previous slide, but I want to like kind of teach it from this slide, and then I'll show you another slide that actually shows you the molecules, so you can kind of start building that kind of chemical view of things. Anyways, here's our 3-carbon sugar, that pyruvate that we got from glycolysis. And again, there's 2 molecules of this, so you got to think that this is happening times 2. So sometimes you'll see this kind of lumped all together. Technically, pyruvate oxidation is separate from the citric acid cycle. You could get some troll kind of questions maybe on the AP exam where they like try and tease that difference out between you. So don't get confused by that. Technically, this is separate from this. In pyruvate oxidation, it's just one step. What we're going to do is we're going to remove one of our carbon atoms in the form of CO2. So one of the products of pyruvate oxidation, and you're going to make more, you're going to make 2 additional CO2s here, is carbon dioxide. So this 3-carbon sugar becomes a 2-carbon sugar, and we attach CoA. CoA stands for coenzyme A. Remember, coenzymes, they help enzymes be more efficient. Basically, it's this molecule attached to this 2-carbon sugar. And truthfully, I don't even really know what exactly it kind of does here. It's not super important for like your purposes. Just know the CoA gets attached to the acetyl. This 2-carbon sugar is now called an acetyl group. That's where that's coming. It's just referring to the 2-carbon sugar. So we attach the CoA to our pyruvate, removing CO2. When we do this reaction, this is an oxidation reaction. You know it's an oxidation reaction because you're taking NAD+, you're oxidizing this 3-carbon sugar further to make NADH, and then you have that H+. And again, all of these H+, anywhere you see the H+ being made, keep in mind those all get fed into oxidative phosphorylation, as well as the NADHs. The NADHs and the H+ all end up going into oxidative phosphorylation. Just kind of keep that in the back of your mind. So we're making NADH here. Then that acetyl-CA goes in the citric acid cycle. Again, this is also called the Krebs cycle. Krebs is the scientist that discovered it.
That was his name?
That was his name.
His parents hated him.
Krebs. Well, that was the last name.
Oh.
His first name wasn't Krebs. That was his rap name. They called him Krebs. Okay. Acetyl-CA goes to the citric acid cycle. We remove the CoA immediately as soon as it goes in. We're we'll release 2 CO2s. You're then going to create 2 more NADHs. And then this is where we're going to get into FADH2. So here we create an ATP. And then we create another NADH. So if you're keeping track, you have 1, 2. So pyruvate oxidation, you make 1 NADH. Again, this is technically separate from the citric acid cycle. So in the citric acid cycle, you create 3 NADHs, 1 FADH2, and 1 ATP. But that is for each time you do the citric acid cycle. Remember, there are 2 molecules of glucose. So this is all happening times 2. So you're actually creating in the citric acid cycle 6 NADHs, 2 FADH2s, and then 2 ATPs. Okay? So the 2's come in, 2, 2, but you have 6 now. So you had 2 NADHs made in glycolysis. You make 1 in pyruvate oxidation, and then you make 6 more in the Krebs cycle. And now we're in the mitochondria here. Notice where we're located. We're in the mitochondria. This is technically the matrix of the mitochondria. Remember, the matrix is like basically like the cytoplasm of the mitochondria. All right, so this is kind of what I really want you to understand. This is a good kind of summary slide right here for you. Get it erased. Very key thing. If you look at that first bullet point, we only do the citric acid cycle, meaning we only go into doing pyruvate oxidation and then the citric acid cycle if oxygen is present. Okay, so if you're a eukaryotic cell, which we are, you have to have oxygen present to do that next slide, the pyruvate oxidation and the citric acid cycle. Okay. CO2 is released. Pyruvate goes to converted acetyl enzyme CoA. The acetyl CoA goes in the citric acid cycle, and then we're going to make 1 NADH per pyruvate, so a total of 2 NADHs per molecule. That's what they were saying right here. They're referring to this process right here. So 1 CO2 there. So pyruvate oxidation, you get out 1 CO2, 1 NADH, and again, this happens twice, so times 2. So you get out 2 CO2s and 2 NADHs. Whereas here, I forgot to add the CO2s. You get out 2 CO2s per turn, so you get out 4 CO2s. Oh yeah, kind of the helpful way, like you could rewrite this as like 6, 4, 2, 2. So 6 NADH, 4 CO2, 2 ATP, 2 FADH2s. Okay. All right. Should have said everything here. Keep in mind, this is all oxidation. We're creating these NADHs and the FADH2. They are electron carriers. The main goal is to take our carbon sugar and keep oxidizing, keep removing those electrons to attach them to the NADH and the FADH2, and then those are what are going to get sent to the electron transport chain. Yep, and then there's that right there for you. Questions? Just showing you where we're at. Now this is where it's in more, yeah, Thomas. Twice per, yeah. Good. So cycle turns twice per glucose. It's per glucose because we took that glucose, which is a 6-carbon sugar, and you made 2 pyruvates. So that's why it goes twice. Glucose is a 6-carbon sugar. You broke it down into 2 3-carbon sugars called pyruvate. Okay. All right. Now this is where, if you take biochemistry in college, they make you memorize all the details. You don't have to know it to this level of detail. You just need to know inputs, outputs. But I think it's worth kind of briefly kind of talking through what's happening. I mean, here's what acetyl CoA looks like. That's the CoA group. This is the acetyl group, the 2 carbons. Something that is kind of worth kind of looking at is they color code these carbons. So these 2 carbons are up top here. And then this 1 carbon in blue, that's going to be these 2 carbons in blue. Those are the 2 CO2s those are the CO2s that get released. So, in other words, you can kind of track these carbons. So these 2 carbons are eventually released as CO2s. And then these 2 carbons, they end up staying with the molecule here. Yeah, here's a redox reaction where you're making the NADH. Here's another redox reaction. Here's another redox reaction, but now you're making the FADH2. So there you go. Any questions? Okay. I think. Let me make sure we said everything on this. Yeah, I mean it's a cycle because then this oxaloacetate, that will then go back in, and then this will meet up with the acetyl CoA, reforming this whole process. So it's kind of interesting as you take this 4-carbon sugar, join it with a 2-carbon sugar, making a 6-carbon sugar, and then you go 2-carbon combines with 4-carbon, making a 6-carbon sugar, and then we go here, we're going to release CO2, so now we drop to a 5-carbon sugar. We lost another CO2, so now we're a 4-carbon sugar, 4-carbon, 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 and you don't become 6-carbon again until you meet back up with the acetyl CoA. Okay. All right. Make sure I said everything here. Again, don't lose sight of what we're doing here. The NADH and FADH2, whether we made them in glycolysis, pyruvate oxidation, or the citric acid cycle, those are all going towards the electron transport chain. That's what we're going to talk about next class. Okay, so that's it for 7.3.